So, as for today, I'm just going to quickly go through the hormonal control topic. We start by talking about the principles and general facts about hormones, how they're transported, what they're made of, and why it is that they can be released into your blood, as a hint, and only affect some of your cells. We will then talk about a specific example, which is insulin control. The video goes into slightly more detail than is required, but I will clarify this at the end. So, a little bit of retrieval practice. You can pause the video and think about these for 6 questions. What is biological control and what environmental issues it used to overcome? Describe how to sample low grazing plants and forest flora invertebrates. Give an error for this and how to minimize it, naming 2 enzyme types, controlled reactions, and giving examples, describing fermentation in animals, and giving 3 functions of the neuron and reflex actions. Okay, so biological control is a means of controlling pest populations. It's the biological side of not having to use pesticides. Low grazing plants and forest flora invertebrates, you probably use something like a quadrat. The error with that would be is if you threw the quadrat to the areas you were interested in. So you have to throw it randomly. That's how you reduce the error. The 2 types of enzyme controlled reaction examples. So that would be your build-up synthesis reactions and your breakdown degradation reactions. You don't need named examples like starch being broken down by amylase into maltose or glucose 1-phosphate being made into starch, but those are 2. Fermentation in animals, so glucose is broken to pyruvate with the production of 2 ATP. That pyruvate is then turned into lactate in the absence of oxygen. No further ATP is produced, so in fermentation in animals you have 2 ATP overall. 3 types of neuron would be sensory, inter and motor, and reflex actions. Explain reflex actions. So reflex actions are protective, they are rapid, and they are automatic, so you don't need to think about them. Reflexes short circuit the sensory cells, so when the sensory cells are stimulated in a way that they feel is dangerous, they will send the message up to the interneurons, to the sensory neurons, and as well as the message being sent to the central nervous system brain, a message will immediately be sent to the motor neuron to have an effect come about, and that's protective. So not all communication happens via electrical impulses. So we talked about electrical impulses being how nerves are transmitted, and then when they get to synapses, there's this neurotransmitter release. So it goes from electrical, and then to cross the synapse, you go chemical, and then we're back to electrical. Hormones are another example of a chemical messenger. So that's the type of messenger they are. It's a common question in the exam. What type of communication is nerves? It's electrical impulses. Synapses is hormonal, and chemical, and hormones are chemical as well. So we're going to look at endocrines and hormones. So, as well as the nervous system, we have hormones. So they are chemical messengers made up of proteins. So remember your share from the protein section. Structural, hormonal, antibody, something beginning with R, which I'll come back to, I'm sure, and antibody, receptor, and enzyme. That's what it is. So, these hormones are made by what are called endocrine glands. So endocrine just means hormone. So an endocrine gland makes a hormone, and they travel in the bloodstream to specific target tissues. Like all proteins that we learn about, it's one half of a jigsaw puzzle. So one half is the male end. It has a wee sticky outy protein, and the other half is the female end. So it has a recess where the sticky outy protein is specific in shape and chemistry. So this hormone, for example, testosterone, is traveling around in the blood. It is bumping off all the cells on the way, but that's fine. Testosterone is only able to affect the cells where when it bumps off them, the wee sticky outy protein on the testosterone fits into a recess in the target cell. So that's how it works. Only the target tissues have the specific receptor proteins that match the membrane proteins on that hormone. So that's the way to do it. Specific membrane proteins that match that hormone. So you can travel all around the blood and only affect the tissues that you're actually trying to go for. So the hypothalamus is an endocrine gland. It's a big one. Pituitary, endocrine, parathyroid, so your thyroxines. You've also got a thyroid gland, things like thyroxines. Adrenal glands, which are things like cortisol and adrenaline. Your pancreas, which is one we'll talk about. And then the gonadal-hormones, so your ovarian hormones such as progesterone, estrogen, and your testes, which produce things like testosterone. And there's other ones that we could talk about, but we shan't. This is a very helpful table because you don't need to know any examples except for the names examples in the diabetes section. So sugar control, you do need to know about insulin, and you do need to know about glucagon. You need to know about the pancreas and the liver. However, you don't need to know about antidiuretic hormone, thyroxine, adrenaline, and in this section, you don't need testosterone or estrogen. So to fill in that table of your booklet, you might as well pause the video and use this. Robert Waldo is the world's tallest man ever. He was 8'11", so almost 9 foot tall. As a result of an overproduction of growth hormone, he weighed in at 35 stone but died at 22. Pituitary gigantism, which is probably what this is, actually runs in my family on my mother's side. So you get an overproduction of growth hormone and you get very tall people who quite often don't live very long. So we'll look at glucose regulation. We've talked already about endocrine glands producing hormones, hormones being a chemical messenger made of protein, only affecting target tissues because the hormone will fit specifically into the membrane receptor of only the target tissues. Blood glucose regulation, and we will go into a little of what's called negative feedback, so how physically these hormones actually work on a chemical level. You don't need to know about negative feedback. It's just a very cute little way of getting you to understand the concept. So glucose concentration in the blood should be kept at the normal limits. If we were in class, in school, I would bring in a blood glucose machine and I would show you where my blood sugar would be. It would probably be at 4 millimoles per litre. It can go up to something like 13. Out with these ranges, it probably means either you've got an insuloma, if you don't have diabetes and your blood glucose level is under 4, or you maybe have some form of diabetes if it's above 13. So we have receptor cells in the pancreas, we have the hormones insulin and glucagon, and we have the effector tissue, which is the liver. So we talked before about motor cells having effectors, and the liver is the effector. So here we go. Eating food, your body will break the carbohydrates down into glucose. Glucose is needed for respiration. You need a constant supply, but too much glucose in your blood makes you prone to infections. And if you use all your glucose up, you don't have any glucose 10 minutes later. So you need to sometimes store it. The glucose concentration in your blood is detected by the pancreas. It has receptor cells on the outside that are constantly saying how much blood sugar has Mr. Collier got in his blood. And that will result in either the production of the hormone, insulin, or glucagon. If your sugar concentration goes too high, your glucose concentration in the blood goes too high, your pancreas will release insulin. So what that does is it causes the cells of your body to be able to take in sugar, so in for insulin, so it takes the sugar out of your blood. And if you think about it, if your sugar level is high, your body produces insulin, the sugar comes out of the blood into the cells, then the blood sugar level is going to drop. The body also uses this mechanism to store extra glucose. So if you've got tons of glucose and you don't just need to get the sugar into the cells, you can store it. So the insulin is produced. When sugar and insulin get to the the liver, the insulin unlocks the liver and allows glucose into the liver. Once it's in the liver will turn the glucose into something called glycogen. So glycogen. You can see it written here with a red E and a red E for liver. So liver and glycogen. This stores the sugar. It's not really important why it is. Glucose needs to turn into glycogen, but essentially, your body is full of water. Glucose dissolves in water. It would be a great idea if you could just put glucose in the liver and it stayed there. But what happens is you go to all the handful of producing insulin, you cunningly put the glucose in the liver, you go, aha, fixed it. The glucose dissolves in water and flows right back out your liver again, which is a bit irritating. So instead what you do is you put it into the liver, you keep one hand on it, you turn it into glycogen, and then it can't dissolve in the water anymore. So that's why glycogen exists as a storage carbohydrate, because without it, your glucose would just run around your body willy-nilly whenever it wanted. But it can't do that if you make it insoluble, essentially. So glucose is turned into glycogen, and if you have too much glucose in your blood, this will get it lower. So it'll bring it down to your 4. If you miss a meal or do lots of exercise, or just normally use up your sugar in your blood, it will start to slowly drop. So this is detected by a different part of the pancreas. So you've got alpha and beta cells in your Langerhans and things. And it will say, hey, wait a minute, I've not got enough sugar. Oh, thank God, earlier on, I stored some sugar as, oh, that's interesting. I stored some sugar as glycogen. So your pancreas says, it's okay, I can sort this. I will produce the hormone glucagon. So it's not producing insulin, because that would put more sugar into the cells and it would drop the sugar concentration in your blood. It produces glucagon. So when glucose has gone from your blood, produce glucagon. So glucagon is produced, it goes to the liver and it says, hey, could I have some of that glucose back? And the liver says, well, you have to give me a second. I turned that glucose into glycogen, so it didn't dissolve in the water and run away. So your liver turns the glycogen back to glucose, and then we talked about it before, that flows out of your liver into your blood, and it slowly raises the sugar concentration in your blood. So glycogen, using the hormone glucagon, is converted back to glucose. You can see all these things that begin essentially phonetically with GLU or GLY get a bit confusing. So this is where spelling, unfortunately, matters. Most parts of the National 5 course, you can get away with misspelling things, and as long as it looks or sounds right, you're okay. Glucose, glucagon, glycogen, glucose, all these things, they sound very, very similar. So if you misspell one of these at any point in the National 5 exam, it is conceivable that you don't know anything about biology and meant to write one of the other ones. So you need to write these correctly in the exam. So if you have too much sugar in your blood, it is put into the liver by insulin, which is released by your pancreas. In your liver, the glucose is stored as glycogen. If, shortly after, you find you do not have enough sugar in your blood, this is detected by the pancreas, the glucose has gone, so it will produce glucagon. That will go and knock on the liver. It will bond to the membrane receptors and say, hey, could I have some glucose back? The liver will convert the glycogen to glucose, and jobs are good, and there's glucose going into your blood. It's a very simple process. So this is the negative feedback thing that specifically you do not need to know, but it helps to understand what's going on. You have a normal blood sugar concentration, 90 milligrams per 100 mils, or about 4 millimole per litre. Increase in blood glucose because you ate a big bar of chocolate behind your filing cabinet. The receptors in the pancreas will sense this. They'll produce more insulin and less glucagon because it's a constant battle, what's called antagonistic action, which you'll need to know about. So extra glucose stored as glycogen in the liver, and we go back to normal. But maybe actually we put too much sugar into the liver. Maybe we pumped ourselves full of insulin, and after enough glucose was put into the liver as glycogen, we dropped too low, maybe to 70 milligrams per 100 ml. So your pancreas notices this, grumbles a bit, and releases glucagon. So less insulin, more glucagon. Glycogen's converted back to liver. But oh no, you went too far. Now you need to produce insulin. Oh no, you went too far. Now you need to produce glucagon. It is a never-ending process of producing more of this, less than that, more of this, less of that. People who have diabetes, type 1 diabetics, their body doesn't produce insulin, so you can imagine what will happen there. They eat too much sugar, nothing happens. They eat more sugar, nothing happens. The sugar concentration rises, they get thirstier, hungrier, tireder, and then eventually they hopefully get a blood glucose level tested and are put onto medicines. Type 2 diabetes, you're producing insulin, but your body's just not paying attention. Those membrane receptors are just damaged or not listening. So, types of signal: nervous was electrical, hormonal was chemical. Transmission of the signal by nerves and neurons in the nervous, and by the bloodstream and hormonal. The things that are affected by nervous can be muscles or glands. In hormonal, it tends to be target cells with complementary receptors. Types of response to muscle contraction, secretion of hormones. So quite often the nervous system sets off the hormonal cascade. Hormonal response is just to produce hormones. So it causes a chemical change in the cell. Speed of response, very rapid. Hormones tend to be slower, but they can hit a lot more parts of the body. Duration of response, so that nervous is short and sharp, so until it stops sending the signal. Hormonal, if you put enough in the blood, it will keep going around hitting those receptors until it is broken down. This is just, I thought it would be useful to put in a slide that shows what the SQA want you to know. The endocrine glands release hormones into the blood. Hormones are chemical messengers. Target tissue has cells with complementary receptor proteins for specific hormones. So testosterone has different membrane receptors to estrogen or FSH or LH or ICSH or whatever. So only the tissue will be affected by this hormone that has the receptors. So you need to be able to name and the locations of individual redo. The names and locations of individual endocrine glands, other than those mentioned in the course, like pancreas and later on testes and ovaries, are not required. Blood glucose regulation, you need to know what insulin does, so it tells your liver to store glucose as glycogen. You need to know what glucagon does, it tells your liver to give up the glycogen as glucose and put it into the blood. You need to know what glycogen does, it stores glucose. You need to know the pancreas is what senses the changes and releases glucagon and insulin, and you need to know that the liver stores the glucose as glucagon and then releases it again into the blood. Detail of negative feedback is not required. So have we look at this, see if you can manage the question. Name an organ that produces insulin, and using your knowledge of respiration, explain why a person suffering from diabetes might be really tired. Okay, the answers to this don't come up, that's a shame. So the pancreas produces insulin. We know that diabetics, from what I've said, don't have the insulin, so the sugar can't enter the body. Glucose is required for respiration, so you won't have enough respiration happening. So not enough ATP, so not enough energy. It's a 1 marker, so you can garble your way through that. Oh, good, maths. Using the information given, which of these is correct? Person A always stays within the normal range. Let's have a look. Normal range is 82 to 110. Person A goes out with 110 because they're way out here. So person B was out of the normal range 180 minutes after eating. So person B, 180 minutes after eating, was out of the normal range because they're above 110. So I think B is correct. B had twice the level of A at 180. Well, no, because 2 times that is 180, so that's wrong. And person A and B both had the highest levels 90 minutes after eating. So 90 minutes, that goes to 190. That is its highest level, but it's not its highest level. So the answer is B. Which of the following identifies X and Y? X, liver. Well, you probably need more information to answer that question, don't you? If we imagined it was, so which of these produces the thing on the right? So the liver doesn't make insulin, liver doesn't make glucagon, pancreas does make insulin, pancreas makes glucagon. So we don't know, but it could be one of these. Specialized cells allow organ Z to respond to hormone Y. This is because the surface cells in organ Z have complementary receptors. Okay, so what I'm going to do is, not that one. Yeah, so we have a Google form quiz that I've put up for you to do here. So you can work your way through that. It's 6 marks. So hopefully you can have a go at that. And the other thing is to do the notes, which I had put up but have disappeared. Where are we? So the notes, as I say, they go through what we have just covered. Just vamping for time here. They go through what we've just covered and hopefully will allow you to understand the topic fully. So this is that table I said you don't really need to know anything except for the insulin line. This is a summary of blood sugar control with a nice picture. This is glucose, probably as we know becoming glycogen, glycogen probably as we know becoming glucose. And we should know that endocrine glands release hormones, that hormones are chemical messengers, that hormones affect only the target organs that have the receptor proteins for them. The receptor proteins are specific to only that target hormone, and how insulin, glucagon, pancreas, and liver control your glucose levels. Hopefully that makes sense. We've also got the meet to do as well, so please do post any questions you have. Come to the meet because you have to, unfortunately, and answer any questions.